Thank you for joining us for our Microplastics Monitoring Subcommittee Kickoff meeting. My name is Erick Burres. I'm with the State Water Resources Control Board. Our co-facilitators are our meeting organizers, Shelly and Scott Coffin. Today's webinar is part of the Trash Monitoring webinar series. This is our last meeting for the series. So today's webinar, as well as the series, has been sponsored by two working groups under the California Water Quality Monitoring Council, the Trash Monitoring Workgroup and the California Water Quality Monitoring Collaboration Network. I am the facilitator for the Collaboration Network. I'm also the coordinator for the Clean Water team. So we've been able to put these webinars together for this past year. And I really want to thank all the folks that have contributed. The goals for this monitoring webinar series was to share our current collective knowledge to support current practices and advances in trash monitoring, as well as the development of data analysis and visualization tools so that we can assess the effectiveness of our current policies and practices to limit trash in our environment. And we've had fantastic presentations and some wonderful presenters sharing with us their information, their knowledge, and how they're running their programs. And if you missed any presentation, want to share a presentation, we have everything available for you. All of our webinars have been recorded, edited, and are archived in a playlist that you can watch on YouTube. So this is today's agenda. I want to thank Shelly for helping me put together our webinar series, and Shelly and Scott putting today's agenda together. It's going to be a fantastic meeting. I'm looking forward to seeing this new working group under the Trash Monitoring Workgroup form. Thank you, Shelly. Thank you, Scott.
Yeah, thank you, Eric. I just wanted to say a brief, give a little bit of a brief introduction to the Trash Monitoring Workgroup. We are under the Water Quality Monitoring Council for California, and we are working on trash issues, and we have three subgroups currently, one on encampments, one on data management and analysis, and another one on citizen science. And we're looking forward to adding a microplastics group as well. I'll give you more information shortly about the Trash Monitoring Workgroup and how you can become involved. We don't want to be exclusive. We want people from outside of California to participate, and I'll give you information on how you can do that at the very end. Scott Coffin is with the State Water Board, and he is all about microplastics. He's going to tell you some of the information about what they've been working on in the State Water Board. But I'll hand it over to him so he can introduce the first group and get going with the program.
Thank you, Shelly. I'm so excited for this. I think this is going to be a really great opportunity for us to leverage each other's resources and work together in this new collaborative space around many shared interests and goals with microplastics. So we're going to kick it off with some management needs and goals presentations from different state agency or different agencies in general. And so our first speaker is Harry Allen. And Harry has over 20 years of operations experience in large-scale multi-agency responses to major natural disasters, hazardous waste sites, oil spills, and other pollution incidents. Areas of expertise include environmental study design, contamination estimation, and application of technology solutions to pollution monitoring and remediation. Harry's primary research interest is in studying the extent of microplastic particles in the environment. He oversaw the development of methods and practices for microplastic sample collection and analysis with the ASTM D1906 subcommittee. He designed and implemented several field studies collecting microplastic particles in marine and estuarine environments, including at Tern Island, within the Papahanaumokuakea Marine National Monument. And perhaps Harry can tell me how to pronounce that. Harry has a BS from Rutgers University and a MS in Environmental Management from the University of San Francisco, where he served as part-time faculty. And Harry also works for the US EPA Region 9. So with that, I would like to hand it over to Harry, and if you can share your slides, please.
Thank you so much, Scott. I appreciate you sharing all that information. Sometimes the bios can seem long, but I think this is kind of relevant because how does an emergency response person become a microplastics aficionado? One of the answers is Anna Marie Cook, who came to my office years ago and said, you know, this is a really urgent need. We need to be working on it. And so if you know her, this is an homage to her in part. But also Superfund Emergency Response is a unique thing, I think, in the world, really. And we have offices all around the country. This is Region 9, so we cover California, Nevada, Arizona. We also cover Hawaii, Guam, American Samoa, Saipan, and some of the other trust territories. We work a lot with territorial governments, tribal governments, obviously the state of California quite a lot, with different departments, wildlife, as well as water board, as well as toxics, as well as radiological. And so we do these decision-based cleanup estimations. So we're on call. Anybody can call with any problem. And if the situation rises to it, we'll do an investigation. So those investigations are not unlike small research studies, but they're decision-based. So we're trying to see if we're above or below a threshold for taking action. And so in the microplastics realm, how many particles is too much? Just like the water board is wrestling with now. We use a simple rubric of, risk is exposure times toxicity. In Superfund, we tend to take the toxicity for granted, and we really emphasize estimating pollutants in the environment. So my whole world is really around field sampling and analyzing that information. So typically these are small studies, but every so often there's a need to look to other places to find the background or the baseline information. And I know all of you are wrestling with that. And as a result, as an emergency responder, I've come into contact with different programs, tribal programs sometimes, who do water monitoring, for example, territorial programs, certainly in Guam, for example, where they do regular monitoring for beaches. In oil spills, I've used the National Coastline Condition Assessment background data. In mine sites, we've used background surveys of what pollutant loads are in water. And so all of this is to say some of the context from EPA comes out of our Superfund investigation of this problem, and it spreads to programs like the Water Division's Total Maximum Daily Load program. And indeed, last summer, there was a effort to put Turn Island in the Papahana-Oumukuakea Marine National Monument on the TMDL list as an impaired waterway. Now, funny enough, when that happened, Keith Kawaoka, who is a UCLA grad and the director of Hawaii's Department of Health, he was on National Public Radio saying, well, I hope EPA helps us because I have no idea how we're going to implement a microplastics TMDL for a remote island. Well, EPA Region 9 happened to already be studying the island for the purposes of cleanup of PCB waste, et cetera. And we were able to do some of this microplastic monitoring in part during that practice. And so to serve these clients, our TMDL users, our territorial governments, our tribal governments, interestingly, several California tribes have robust water quality monitoring programs. You may or may not be aware of that. But a lot of these people are doing practices and doing routine information collection. And what's a good way that we can tie that information to upstream management decisions? Here's my favorite example. I live in Los Angeles now in this particular watershed. And yeah, it's quite messy. It's Shelly Moore knows. You see you nodding your head. It's quite messy. It's a playground for somebody like me who likes to clean up stuff. I mean, wow, it's quite a lot to handle in just this one watershed. And I really appreciate some of the comments I've heard about watersheds. I see Diana and Scott there. And studying those watersheds is really what's important. for understanding what to do about it. We can't make blanket decisions without really checking how those decisions and those things are performing, just like when we clean up mines to satisfy a TMDL for mercury or lead. So how are we going to do that? For me, interestingly, I did a parallel process to the trash monitoring playbook, not knowing about it. I made my own app. I drove all around LA taking pictures because I go all around LA for other reasons. And I have my own map. So I kind of put that side by side with the Southern California bite sampling locations. So just like this, how do you take those two disparate pieces of information and combine them? And with that, how do you pick a good TMDL? If EPA was going to select a TMDL measuring method for Turn Island or for LA, should it be trash playbook style or should it be microplastics playbook style or both? We don't have the answers to that yet. And so this is why it's such a compelling problem. But we are working on methods for the microplastic side. And I am familiar with the trash playbook, which I think is excellent. I want to see how these two things can interact. We at EPA, that's Anna Marie and Bill, we worked with MBARI earlier on and Monterey Bay Aquarium Research Institute to come up with a microplastics sampler. And also we've worked quite a lot with the Department of Public Health. Jeff Wagner, who's on today, we've been working with him since about 2011 on microplastic sensing because we worked with him on other emergency type pollution incidents. So how can we use particle sampling and a combination of mass-based and count-based information to inform decisions? So in addition to collection, which we've done at wastewater plants and also open water, we've got a pretty robust sample preparation practice, which many of you are familiar with, how to prepare a sample for analysis in the lab. We recently got through our reference sample practice, which Jeff may talk about, how to prepare a target sample for quality control. We are balloting a pyrolysis GCMS analytical method. It received a negative. It's a consensus-based process. It's very difficult. Got to go back and address that negative, and hopefully we'll reballot that. And then with Jeff Wagner's help and many others, we're doing an FTIR analytical method for ASTM. All these are interesting to me, and I'm finding I'm a budding chemist after all these years. But really, my expertise is in how many samples and where, and can we really get the resources and attention we need to go back to the same place? So that's the difference between sampling and monitoring. Sampling is the one-time shot, at least this is EPA lingo. It's the one-time shot. Monitoring is repeated sampling or repeated sensing, such as the Southern California Bite Study or the SFEI, regional monitoring program. These monitoring programs are really what's needed. And so when you're designing a study, there's different kinds of things you need to consider for those things, not just the analytical method, but importantly, the study goals. I listed a few here, which may be unfamiliar or maybe outside of the box. Some of those study goals, some of the interpretation of that information, and so on. And I'm really interested in using EPA's quality assurance program to drive decisions around this. So I'll conclude there just really quickly. EPA's quality assurance program's got a lot of this built in, and there's a lot of standardized analytical techniques, et cetera, to be used. And I hope to bring that to people's attention and make it more well known. Thank you.
Thank you, Harry. I want to highlight, first off, two pieces of legislation that are mandating the State Water Board to take action on microplastics. The first is Senate Bill 1263, which requires the State Water Board to work with the Ocean Protection Council to initiate a statewide microplastic strategy by the end of this year. And by 2026, we're required to develop a risk assessment framework, get some standardized methods, establish baseline occurrence data in the environment, investigate sources and pathways into the marine environment, and go back to the legislature with recommendations for source reduction. We have a similar mandate on the drinking water side. California Senate Bill 1422 required the State Water Board to first define microplastics in July 1st of last year. And by July 1st of this year, we're required to adopt standardized methods for monitoring in drinking water, a 4-year plan for testing and analysis, consider adopting a health-based guidance level to aid consumers in interpreting what microplastics in drinking water means, and provide accreditation for laboratories so that we can be sure about the results. So we've been conducting a lot of research in order to fulfill both of these goals in the past 2 years. And I want to give you the highlights of this is really what we've identified to be the monitoring needs and the method needs for both the environment and drinking water based on our toxicology research as well as the method development. So for the aquatic environment, we've got a lot of different matrices that we're going to need to apply methods for, including high and low turbidity, such as wastewater, ocean water, stormwater, even fresh water, as well as looking into sediment and animal tissues. And the majority of these samples don't need to be super small particles. We can get by with a lot of mannitol data and larger particle characterization. And I'll explain why in a second. We will want some samples that are as small as we can get, below these 212 micron grab samples. And our desired detection limits are same for aquatic and drinking water. It's about 1 particle per liter, between 1 and 5,000 micron particle sizes. And I'll explain a little bit more about that in a second, but this is based on toxicology. For drinking water, much more narrowly focused on the source water and the treated water. And for source waters, we can get by with larger detection or larger particle sizes. But for treated water, we really have to get to the smallest particles we can. And I'll explain why in a second as well. So to give you a little bit more context, we had a health workshop that was conducted from October of last year and just wrapped up. This was a group of experts that evaluated the literature and derived ecosystem thresholds where we would expect to see different levels of harm to the environment, and considered adopting drinking water thresholds, developed a framework for assessing evidence for mammalian impacts. So I want to share a couple of insights that are relevant to the monitoring world. The first is that we identified that for ecotoxicology, the sizes that are being conducted, or sorry, the sizes of particles that researchers are doing laboratory experiments on are dramatically different than what they're actually monitoring in the environment. This is a significant mismatch, and we want to close this gap as much as we can. But there are a lot of analytical limitations, feasibility limitations that stand in the way. So as much as we'd like to get down into the very, very small particle sizes, it's quite challenging. However, if we know the particle distribution in the environment, it may not matter. If you have, for example, a well-characterized distribution of particles by size in the marine environment shown here, and you take a grab sample 20 to 212 microns, then you can reliably extrapolate the number of particles that you would see if you had sampled within the entire size band that you're looking for. The particle size distributions are known to be compartment specific. These depend on the different parameters in the compartment. So for instance, the exponent for the distribution is different from biota and surface water, and you can break it up by marine and freshwater. And the data that I'm showing you here is based on some sites in the Netherlands. And this type of analysis has not been done in California yet. So we really want to apply these sophisticated methods that can get high resolution particle data down to a very small size, around 1 micron or so, for some samples in the marine and freshwater environments, so we can make these reliable extrapolations. For drinking water, it's a little bit different because we have treatment. So we can do these extrapolations for source waters that are untreated, but when you have treated water, you're removing most of the particles that are above 20 microns in size. So we really need methods that can get reliable detection for those very small particles if we're to look in treated water. Also, we just wrapped up our method interlaboratory validation study. This included Raman and infrared spectroscopy methods for drinking water, and we're also evaluating these for some additional matrices. And the methods are available now on the State Water Board webpage. We just made them available yesterday, and we're looking forward to getting stakeholder feedback on these methods. They have been harmonized through an formal interlaboratory comparison study with 22 labs in 6 different countries, and we look forward to seeing them be used more by the community. Really briefly, we have some idea about the different pros and cons about these methods. In general, FTIR and Raman are very good and reliable methods that can get us down to about 20 microns or so. Smaller if you have more sophisticated techniques. But the biggest thing to note here is that the measurement time and the cost associated with that is rather high for an analytical method. So we need to improve this and find shortcut methods. And you'll be hearing from Terri Slifko later about more details on that. So really briefly, in terms of what the State Water Board is doing on the drinking water side, we are required to adopt a 4-year plan for sampling and monitoring by the end of this year. And for what we're thinking right now is that this will be an iterative approach, where the first phase, we're just focused on characterizing the sources. And in an additional phase, we'll really narrow in on where we believe to be some problems could be occurring. In terms of where we find particles in the source water, we might want to do some monitoring in the treated water. After that 4-year monitoring, we will go back to our typical monitoring, how we monitor typical emerging contaminants. So there's 3 main goals that we have. One is further validating the Raman and infrared methods and making sure that the community can properly implement these methods. We need to get methods with lower size limits of detection that can be rapid and automated. We also need some cheap and rapid surrogate methods. And again, you'll hear from Terri Slifko about that. So with that, I am going to hand it back over to our next speaker. And I believe I'm introducing them. So we've got Rebecca Fitzgerald up next, who manages the Surface Water Standards and Assessment section in the State Water Board's Division of Water Quality. She has been with the water boards for 22 years, mostly at the North Coast Regional Water Quality Control Board, with the last 4 years in her current role at the State Water Board. She manages statewide water quality with control projects, the assessment of data for listing water bodies as impaired, and the TMDL program. She graduated from Sonoma State University with a bachelor's in environmental studies, and you'll be able to find her this weekend in Yosemite Valley if you need to follow up on a conversation.
And I'm looking forward to it too. Thanks, Scott. Thanks, everybody. Wish I could be looking at you all in a real room and not just myself and my notes on a screen. Hopefully, eventually, one of these days we'll get there. I will start by just saying that I'm new to the world of microplastics and very much still on a learning curve, and very much appreciate all the work and the science and the research that has already gone into this pollutant and this issue. I was asked to touch a little bit on management needs and monitoring goals. And there are 3 that I thought I would touch on today, and I will recognize that I am blending monitoring needs with scientific research needs, and then I'll get into some thoughts about where we may go as far as policy options as well. So first, I would say a need that we have is scientific research to help illuminate the best microplastic metric or parameter, with consideration for environmental exposure pathways, what is that size, what is the best parameter when we talk about microplastics that we can use when we start looking about what we're going to do about it. And then following that, data linking that microplastic level with impacts to the beneficial uses of water. I'm seeing more and more come through now as examples about how many particles per volume cause chronic effects, how many particles cause acute effects, and on different types of uses from aquatic life to human uses. certainly recognize that microplastics are a diverse class of contaminants, and that there are different exposure pathways and different risks. And so having more information about those pathways and risks, I think is a need that we still have. Second, I would say we need to continue to define the sampling and analysis methods so that the data that we are using are of high quality. And Scott, you just touched on some of that. I would say third, we also need data that characterize conditions in California waters. Before we do anything as far as following up with any sort of policy, I think we need to know what the status of our water bodies, both marine and bay, and sometimes even inland waters are in terms of microplastics. The science should drive the standard setting and policy decisions work that will likely come. I think we all recognize that. But there's also a point to be made that we should avoid collecting data without considering how the results will be used. And I know that there's always questions about, well, how are we going to use the data that we're going to collect? So I wanted to share just some very early conceptual options that are being considered, at least on the water quality end, for the water boards about how we may use thresholds and data of ambient conditions. So we could use it to list a water body as impaired on the 303D list of impaired water bodies. This often results then in the development of a total maximum daily load or other regulatory actions to control sources. We could consider setting a clear numeric water quality objective for microplastics to protect our beneficial uses in water bodies. Certainly, as we were looking at the statewide ocean plan or water quality control plan and prioritizing what projects we wanted to pick up in the future, we were recognizing that microplastics is an emerging issue and one that we may want to prioritize, and we have prioritized as a high priority, to consider doing a water quality objective. Another options is to use the thresholds and the data that are coming in to consider anti-degradation and potentially use that to identify where we can control sources, the optimum means of controlling them. And we can do that without establishing an objective if we have the right thresholds and data available. We can, once we have an objective or control actions, those can be incorporated into permitting mechanisms. I think that that's certainly something we should be considering and aware of. Objectives and control actions could also be incorporated into focus monitoring for stormwater discharges into areas of special biological significance on our coast. And there are certainly probably more options when it comes to what we could do with microplastics once we have clear thresholds, clear data. And I certainly am looking forward to some of the risk assessment work that is currently underway. I also want to say that we do not yet have a preferred approach, but I do think it's helpful to hear what some options could be. And I'll just wrap up by expressing some appreciation. I know that there's a lot of monitoring efforts and research that has been done to date. I'm certainly looking forward to the rest of this discussion this morning and where our next steps go. I specifically want to acknowledge the work done by SFEI and SCCWRP for the monitoring and assessments of health effects. I really appreciate the effort of OPC and everybody who's working with OPC to develop the statewide microplastic strategy per state SB 1263. I think that's going to be a really helpful tool and a direct benefit for us. So just wanted to conclude with that. Looking forward to the rest of the morning. Thank you, Scott, for reaching out and inviting me. And if you guys have any questions, I'll throw my email in the chat in case you want to reach out.
Thank you, Rebecca. We're going to go to one more speaker for this first session on microplastics management needs and monitoring goals of agencies. And I'd like to introduce Suzanne VanDrunick. Suzanne is the National Program Director for the Safe and Sustainable Water Resources National Research Program in the Office of Research and Development at the US EPA headquarters. The comprehensive water research program includes science and technology research on watersheds, economic benefits of water quality, nutrients, and harmful algal blooms, and water infrastructure and reuse. Suzanne's background is in aquatic toxicology and pathology. And
Thank
Suzanne, I can go ahead and share your slides if you'd still like me to.
Thank you. This will be fairly quick. I only have 2 slides. If you want to go to the second slide, next one. Thank you. You already heard from Harry about some of the microplastic related research that EPA is doing. In ORD, we coordinate with the regions with rare projects, and you had a nice description of that. We're really focused on analytical methods for sediment and surface water, and we've collaborated on the drinking water study that you heard about from Scott. So real briefly, at the present time, EPA doesn't have any specific stated monitoring or management goals for microplastics. However, in Section 301 of the Save Our Seas 2.0 Act, enacted in December of 2020, it directs the federal government agencies to develop a comprehensive study that looks at the U.S. contributions of plastics to the global ocean plastic waste and marine debris. It has a number of things in this act. It directs the study on the effects of microplastics on marine ecosystems and human health. EPA specifically is responsible for developing a public post-consumer materials management and water management strategy. I think I got that right. And at the present time, the agency is considering including an objective on microplastics. So if you could go to slide 2. So this is just a, I'm sorry about this. I'm having, can you hear me okay?
Yeah, we can hear you.
Oh, okay. So this is just a quick overview. Right now, ORD is developing its next 4-year research plans. It goes from fiscal years 23 to 26. And in the process of doing that, we have a list of priorities from internally, the program offices, and also the regions. And we spent a considerable amount of time reaching out to states and tribes, having listening sessions with them to try and understand what their water-related needs are. So this is just a quick snapshot of some of the high priorities related to microplastics that we may be considering moving towards. And this science would support either at this point, non-regulatory, voluntary management and monitoring. So real quickly, so the regions are very interested in impacts on fish and shellfish species, developing standards across various media, standards for assessing using FTIR and Raman, you've heard about that, and tire dust as a source of 6-ppd quinone. The states, they have expressed primary interest in source identification, fate and transport. We hear that a lot. Sampling methods and analytical methods with relevant appropriation detection limits, and Scott did a really nice overview of that. Looking at the impacts again on public health and ecosystems, not just the plastics themselves, but the adsorbed chemicals. And then options for removing microplastics from water, solid waste, and other media. And we also heard from the tribes, similar to a concern expressed by the states, about the use and fate of these tire anti-degradants. One of the most common chemicals is the 6-ppd, but the concern is the quinone metabolite. the byproduct, which is believed to be very toxic to aquatic organisms. So this is kind of a snapshot of the research that we're thinking about, thinking about what we might possibly be able to support in this next 4-year research plan. And that research plan will be final in September of next year. And that's all. If you want to reach me, it's real easy, vandrunick.suzanne@epa.gov.
We have a question that came in through the Q&A. Curious about Suzanne stating EPA's work on FTIR, infrared, and Raman. What about PYGCMS, as Harry Allen mentioned?
They are also doing that work. I think that might be in the Cincinnati lab. So I can follow up and give you a summary of the different approaches that we're using and the different capabilities we have. So there's a number of analytical methods and instrumentation spread across EPA's labs. We have 10 different facilities, but primarily the work is being done in Narragansett and Cincinnati, and it does include those techniques.
Yeah, that's right. The Narragansett lab, I believe, is trying to get an instrument, and the Cincinnati lab is working with us. So they're sharing samples that they collect with us locally in California to do analysis.
Thank you, Harry.
I got a quick comment. So Suzanne mentioned the RARE program, which I think it changed its name to ROAR, I heard. But that's a really interesting program. So basically, EPA in the regions, we partner with, like I said, states, tribes, and territories to do specialty research projects. And so if a partner comes to us wanting to do a project, we find a partner from Suzanne's program, and we make a pitch for a small amount of money. It's usually about $50,000. These projects have been very successful in the microplastic world. So we've done quite a few of them, and all of our ASTM work on the outside has been accomplished through that process. So there's those and other EPA grants, including SBIR and other small business innovation research that are available. And by the way, all of them require quality assurance statements in compliance with the EPA's quality programs, and that's why I'm pitching that.
We also, thank you for mentioning that. We also have the STAR grant program, which I think is fairly well known. And under a different spending authorization, we have national priority grants. And those are very large amounts of money. They range from $4 to $6 million. And probably the average award is roughly about $1 million. So that would be another way that we could fund microplastic research if we don't have the expertise, which in general we have adequate. We have pretty good expertise in EPA, but sometimes we don't have the bandwidth to do the work if there are other priorities to do. So that would be another place to look for when the grants are posted.
Thank you. We have one last question. This is for you, Scott. To what extent do you also look to data analysis methods when developing methods for drinking water monitoring?
So there's a couple different components of the data analysis that are very specific for microplastics analysis, and you could sort of split it into two categories, where one is what the laboratories are doing in-house, and that is some of that is captured in the standard methods that we just reported or that we just released, but some of that is also specific to the instrumentation and data management within the laboratory. And the other component is what we do with the data once we receive it. And the modeling that I showed you using the different probability distributions is just one of the ways that we process that data and compare it in order to make inferences. And so there's quite a few issues going on or there's quite a few things to consider with data analysis, as you can see. Be happy to talk about this more with you at the either offline, or perhaps this could be a breakout group within our monitoring work group.
Thank you, Scott. We're going to move ahead to our second session for today. Next is State of the Science and Research Needs. We have two presenters. After they give their presentation, we may have time for one or two questions. Our first speaker is Jeff Wagner. He is a research scientist at the California Department of Public Health. He received his PhD at the University of North Carolina, Chapel Hill. Go Tar Heels. He's providing subject matter expertise on sampling and analytical methods, study design, and data analysis over there. He also manages a microscopy and special studies laboratory and maintains accredited asbestos analysis programs. Following Jeff will be Charles Wong. He heads the chemistry department at SWRP, the Southern California Coastal Water Research Project. He received his PhD in civil engineering from the University of Minnesota. He joined SWRP in 2019, where his research interests included the development of novel methods for quantifying chemicals of emerging concern. Thank you for being with us this morning, gentlemen. Jeff?
Thank you for the introduction. Can everyone hear me and see my full screen?
Looks great, Jeff.
Okay,
Yes.
great. So I'll be speaking today about precision or repeatability of microplastic analyses. And I just want to start with thanking EPA for funding this work. As Harry mentioned, we've been working with EPA for some years now, and to the other labs that participated in the interlaboratory study. So I want to start with the need to understand variability when comparing two different results. For example, the microplastics concentrations at two different wastewater treatment plants. This is separate from the other critical issue that a lot of people have been focusing on recently, which is also critical, accuracy and bias. But this topic deals more with, you can think of them as error bars on your results. And that's really key when you're trying to decide whether two results are really different. So in practice, this variability is a combination of many different sources. And I have sort of a cartoon at the bottom that tries to show the variability that would come from different steps of the sampling and analysis process about a mean, which is what you're trying to actually get at each step. So for example, when you're sampling, you can picture microplastics concentrations at a given location varying over time, and you're grabbing just a grab sample. Another example would be once you got that sample, if it's a big sample, water sample, when you take an aliquot from that sample, you can imagine there would be variability of the concentrations of all the different possible aliquots from that sample about the mean sample concentration. Similarly, when you're preparing the sample, there could be variability from the preparation process. And then finally, the piece that I'm going to be focusing on today is when you analyze a given prep, would repeat analyses of just that prep vary? And some reasons why that might happen is if you choose different fields or particles to analyze each time, there's a certain amount of signal to noise in your spectra for microscopy, but also for other analytical methods that could contribute variability each time you do it. So in regards to field selection for microscopy methods, that pretty much comes from the fact that when you have heavy concentrations of either microplastics or more often of other things besides microplastics, you'll want to, in many cases, spread your deposit out, say on a filter, to minimize the matrix interference and spread out those particles. And in that case, it's quite common to analyze a fraction less than 1 of your sample. It reduces your analysis time, particularly if you have thousands of particles, or if you have a slower instrument. Sometimes it even improves filter flatness and focus issues. But in any case, because of this, there's many different environmental count-based methods, particularly in air samples where this type of practice is assumed. And therefore in the methods and I should add in the accreditations for these methods, it is always specified that you assess the variability from repeat counts. And I can kind of try to show what this looks like for a few of these tests. And asbestos in air, you're actually subsampling the filter and then from there subsampling that subsample with kind of a systematic field pattern. For asbestos in water or air with TM, the actual analyzed area is much smaller. And then on the other extreme, for microplastic analyses, you typically analyze a much greater proportion of your sample. So what we did was set up a precision exercise to try and assess analytical precision from repeat analyses. And this was targeted at infrared instruments. And we prepared both clean water and wastewater reference samples for this exercise. The way we did this was to spike vials of 10 mils of liquid of either ultra-pure water or treated wastewater in methanol to simulate aliquots from larger samples. And we used microplastic spikes from 50 to 500 microns. I should have put on the slide, we used PE, PET, and polystyrene for this exercise, and we designed it so that they were low concentrations, actually less than 40 microplastics per sample. And we did that to accommodate different methods, some of which you might want to count the whole sample and then recognize that every lab would be doing this 7 times for each sample. So this was a worst case for precision, but it may be relevant to cleaner real-world samples. And this was a relatively small exercise with just 4 labs, more of a proof of concept. We did this to test the draft ASTM methods that Harry mentioned earlier, with the acknowledgement that different instruments possess some differences in workflow and the final sample prep, and that performance may depend on these variations. So this is a look at the data flow. Basically, each lab received a spreadsheet template, simple template where you just record the polymers you identified and their sizes. And that had 7 tabs for the 7 duplicates for each sample, and it automatically calculated the relative standard deviation of the counts for each detected polymer type, as well as the total microplastics. We also, as part of the reporting process, asked labs to give some details on their extraction and analysis methods. And just to point out a few relevant differences here, 3 of the 4 labs did filtrations. 1 did a pipette, series of pipettes onto slides. And that resulted in, that was one of the factors that determined how much of the sample they actually prepped for analysis. It ranged from 100%. Most labs actually did 100% for the clean water sample, but much less for the wastewater sample, just because it had so much remaining material in it and it was difficult to deal with. The pipette, the lab that was doing pipetting prepared even less. And as far as that wastewater samples go, yeah, half the labs found it advantageous to do additional chemical treatments to try and reduce that matrix, and the other half did gravity separations. So on the analysis end, they were all IR techniques, and all of them used a microplastic reference spectra in combination with automatic ID. But they varied in the degree of automation. 2 of the labs had automated mapping techniques with autocorrelation maps. Another one did manual particle selection with automated acquisition, and still another one did fully automated particle targeting. And the last thing I'll mention is that one of the labs attempted to reorient the sample for each duplicate, but then that was just for the clean water sample. And I'll show why in this slide. It turned out that that was not a good idea. Even though you may want to assess that as part of your variability analysis, the orange data, or rather the blue data, represents what happened. So basically, you can see that the microplastics counts went down each time, suggesting that there was particle loss from the sample. So that basically introduces an artifact into this precision analysis. So I would propose that an alternative approach would be to just make sure you analyze different fields each time rather than repositioning the sample. And then the orange data is for the wastewater sample. That was where that second process was followed, and you can see you get a much more stable recount over time. I mentioned that the wastewater samples were heavier, and basically because of that, they were much more time intensive for this interlaboratory study. And certainly the degree of automation that can be incorporated into a process helps with that. This is an example of spectral correlation maps. So in this case, you only have one microplastic particle out of hundreds, and these types of correlation maps can help you just find the one microplastic. So it saves time. Moving on to the results, as I mentioned, these were low count spikes. So the labs had detections ranging from roughly 10 to 30 microplastics per sample. So again, fairly low counts. And we instructed the labs to recount all the polymer particles that they found. We knew there would probably be extra polymers besides the spikes in the wastewater, just native to the wastewater, because these were actual wastewater samples. And then there were also extra polymers detected in the clean water sample, probably during the prep process were introduced. But the key, again, for this exercise was not recovery, but reproducibility. So if you see a particle, just see how many times, see if you see it every time. So moving on to the RSDs, they ranged from about 0.1 to 0.6 for the total microplastic counts. And what we found, you can't really see it from this data, but if you plot it, you can see that, as you might expect, those RSDs were dependent on microplastic counts. So this is sort of showing the same data three different ways. But in the upper leftmost graph, you see that solid black curve is what's predicted from Poisson theory, that if you have low counts, your variability just due to random chance in counting will go up. So that implies that, alternatively, that if you want a low RSD, you're going to want to count more microplastic particles. And that is expressed in this equation on the left. 1 over the square root of the mean count is your expected variability due to chance. And what we found was that our data generally followed that trend, although in many cases there was less variability than what would be predicted. So that was interesting. What our main concern was the opposite. Basically, if we would have found that variability was greater, much greater than random chance, that would have suggested additional sources of imprecision in these analyses, or worst case, just analyses methods that you would call unreliable. But fortunately, we found the opposite, that they did follow this Poisson prediction, and in many cases was better. We found a similar result when you did a different Poisson statistics-based analysis. Basically, this is showing the number of trials in which the mean count was obtained. You're never really going to expect all 7 trials to obtain the same mean count. You'll expect sort of a distribution. And again, we found in more cases than not, it was actually better than predicted due to random chance as far as obtaining the mean count. Doing a statistical test at a confidence level of 0.05 showed that 1/3 of the data sets represented truly random recounts. And this is yet another way of showing this data, where a score of 1 means that it was what you would expect for a random recount of low count samples. And then again, the ones that were not representative of random recounts were generally better than what you would expect from a random recount. So in summary, we feel that we did design and complete a successful precision exercise in which the low microplastic counts and wastewater matrix in particular were challenging, though interestingly, we found that the RSDs were similar between the clean water and wastewater matrix. And we found that the RSDs decreased with increasing counts, which suggests that you should either try to aim for a certain threshold number of counts in your analysis to achieve a certain RSD, or if you have to count low counts, scale your expectations accordingly that you will get a higher RSD. That being said, we found RSDs that were similar or lower than random chance, and we think that the automation in these methods likely improved their reproducibility. And then finally, selecting different fields for each duplicate is preferable to reorienting the preps to avoid artifacts. Now, the implications of this exercise, I just wanted to mention, again, this is just part of the total variability. So additional sampling and subsampling variability assessments are needed. But for just the analytical precision piece alone, I think what could be done in the future is that for a given lab, instrument, method, analyst, and I would argue microplastic range, you can track these things in control charts. So you do it once initially and then update it regularly, say every analysis day, week, or quarter. And you end up with a control chart. I'm showing an example from our lab for our accreditation for asbestos and air. So that generates an RSD that you can report, sort of a characteristic RSD for that lab instrument method analyst that you can put on your reports. It can be multiplied times a given analytical result to give you an analytical uncertainty. And it can also be used as a way of checking real-time your any analytical issues that come up in your lab. So when you do your latest recount, if it's outside of control limits, then you have a reason to investigate problems with your lab. And that's all I have for today. And thank you for having me.
Thank you, Jeff. Next up is Charles.
A pleasure to be here. Thank you all for coming. I was asked to talk a little bit about microplastics analytical development at SCWRP by Scott Coffin, and I'm going to follow up on some of what Scott talked about with regards to our microplastics measurement intercalibration study, which has wrapped up for drinking water and which he has made available as he's mentioned. So, so what is this? This is basically a study to determine what methods would be suitable for monitoring microplastics in drinking water in response to state legislation mandating this. And this is a significant challenge because up until fairly recently, there hasn't been a whole lot of consensus on how to measure microplastics. And that's problematic because, for a bunch of reasons. If you're doing different methods, you could be comparing apples and oranges, and it's hard to intercompare results from data obtained from these methods. It's also not clear what these methods can and cannot do, or if any of these methods are better than others, and also why. And to compound this, it's also not necessarily clear what exactly it is we want to measure with microplastics. Is it their size, their shape, their polymer type? You'll hear more about that in some of the other presentations later on in this webinar. But basically, this is a significant challenge, and to add to this, of course, is that developing these methods often take a long time. You got to figure out what you want. You got to figure out how to go about doing it and what you're after. Then you have to perform various analyses, for example, for a number of different labs to see what the method can do. Look at the results, figure out ways of reducing variability, and then basically repeat the process. So how did we go about doing it for this particular issue, which is to find ways to monitor and measure microplastics in drinking water? Well, The intercalibration study that SCWRP led basically used blind samples and a common playbook, common set of methods that participating labs could basically follow to process and analyze these samples. By blind samples, what I mean is these are samples with known compositions that we've added, but we don't tell what the participating laboratories what's in them. So they analyze these as unknowns, and they follow a agreed upon series of procedures to minimize variability from that. And from this, we're basically quantifying a couple of things. We're quantifying accuracy, we're quantifying precision, and we're quantifying feasibility. So accuracy is basically how close can you come to the middle? What are each lab's results compared to what we know is the correct answer based on all sorts of things. Precision is how close can each lab cluster their shots to each other? How repeatable are they with the triplicate samples that they get? And we can parse this in a number of different ways to figure out what these methods can and can't do from the same laboratory, from experienced laboratories, and from labs with different levels of experience, which is important because in monitoring work, there's going to be laboratories that are new to this, and we want to see basically how well they can do and how they can improve if they do need to improve. The final thing, of course, is feasibility by figuring out, well, how much time and effort and energy does it require to do these types of methods? Because that's important too from the monitoring standpoint. So I want to talk a little bit about some highlights from this particular from this exercise and what would fall out from it. Back in 2019, we had a workshop. We invited experts around the world to figure out some candidate methods, which you see here, and also draft some standard operating procedures, again, so that everybody's following the same playbook and we minimize basically people doing their own thing as a source of variability. Previous intercalibration studies have shown that that's a pretty significant source of variability. With regards to the blind samples that we made, here's a little bit of information about them. There were several different types of polymers that were added. These ranged in size all the way down to single-digit micron size to about a millimeter in size. These are broken up into 4 different size fractions, as you see. We have different types of shapes. We've got pellets and fragments and spheres and fibers. And one of the other things we have to simulate what would be in a real sample would be false positives. So many of the particles that you would expect in an environmental sample, including a drinking water sample, aren't going to be microplastics, but they might look like microplastics. So we decided to add material that would basically would be common that you would find, or at least would look like microplastics, and these would include things like sand and pieces of shells and cotton fibers. We even added some bunny fur for my pet bunnies because oddly enough, that looked an awful lot like the actual polymer fibers that we actually did add. So what are some of the matrices? We have clean water. You can see an example from the nice clean water there. That's complete, and that's what I'm going to talk about. We also had environmental matrices such as a proxy for surface water mannitol, the dirty water, which also included algae and sticks and all that sort of stuff. That's that yellow container you see, as well as things like sediment and fish tissue. That data submission is complete, and we are now starting analyses in conjunction with our participating laboratories on these other matrices. So I'm going to confine my remarks to the clean water matrix. Overall, for all 4 matrices, we had 40 participating laboratories in 6 countries. This was a healthy mix of public sector labs at various levels, academic labs and private sector labs, and they ranged from some of the experts in the field since it started to basically labs that had never done this before, with quite a few labs on each particular method. And basically how the lab flow worked was we had blind samples. You can see a bunch of them being ready to shipped out that would go to the laboratories, and the laboratories would again follow the playbook of the draft SOPs to process these types of samples and then quantify them using visual microscopy, measure them, figure out how many they are. This is a very common step as currently done in microplastics analysis. What this won't do, of course, is verify what the material is and if it's a plastic particle or something else. So there would be a confirmation step using something like Raman or FTIR spectroscopy or pyro-GCMS. With regards to the clean water matrix, which was a proxy for drinking water, the processing method was basically split them apart, split the size, the particle sizes apart in a number of different sieve sizes, put them on filters, and then basically count and quantify. So what kind of results did we get? These are for some of the candidate methods here. These are box plots showing the distribution of what, how many particles each of the labs got. That's the mean of those is each of the individual dots. And you see two dotted lines near the top. The lower dotted line is the total number of plastic particles that we actually added. And the top line is the total particles we added, which also included all the false positives. So if you look at this, you kind of go, well, this doesn't look all that great. More than half the labs got less than half the particles that we actually added, and there's quite a bit of variability involved. But remember, there's also quite a bit of factors under the hood that's we can look at further, and that gives us a lot further insights into what kind of performance these methods have. So what we have plotted here are box plots, and the blue box plot on the left is basically what I showed you in the previous slide, and it's now expressed as relative recovery. But the other box plots now break these down into the 4 different size fractions that we have, the different colors that we have, and the different types of particle shapes that we have. Now you can see there's a lot more going on here. So what you can see here is that with regards to sizes, for anything bigger than about 20 microns or so, as it turns out, the labs actually have pretty good recovery and reasonably tight precision around the actual value. It's the 1 to 20 micron particles, which are very difficult to analyze, and that's known in the literature, that caused all sorts of problems and also was depressing the overall recovery. With regards to things like, let's go to the far right, morphology and the shape, you notice that the labs typically reported a lot more fibers than we actually added. And as it turns out, it's actually very, very easy to contaminate samples because there's particles floating around in the air and on surfaces. So we have to keep things clean and try to do everything you can to minimize contamination. And a lot of the contamination is due to things like fibers. And these would, for example, also include fibers coming from masks from that you would have to wear for the pandemic. With regards to things like color, you can see that things like blue particles tended to be higher. And as it turns out, the blue particles that we did add to these samples tended to be rather fragile and tended to break up. So that's partially what you're seeing there. We can also get quite a bit of information about and insights into best practices. So for example, these plots here show you the recovery for one of the size fractions in terms of both accuracy and precision based on how much experience the labs are reporting. So the blue labs at level 0 have the least amount of experience, and the gray labs at level 2 have the most amount of experience. What we know here is that the least experienced lab tended to overestimate the number of particles that they have, and they tended to be pretty volatile in terms of identifying plastic particles that weren't, and vice versa. As you gain more experience, the recovery, you stopped overestimating the number of particles. You started to figure out what a plastic particle looks like and confirm that that is indeed plastic. And basically you weren't all over the map and trying to identify them. So this is good news from the standpoint that once you learn how to do this, you're going to get better, and measurably so. What we also learned is that training and following a clear SOP also helps. So we had a training course back in the fall of 19 where a number of labs went to be trained hands-on on how to do these types of extraction processes and also measure these things. And those labs that did the training got better performance. Also, those labs which followed the draft SOP that we offered, the common playbook, also had better performance than those labs that didn't. And this is important from the standpoint in that a lot of labs doing monitoring aren't going to be familiar with this, but if they are trained on how to do this, and if they have good, clear SOPs to follow, they can get good performance and they can improve their performance. And with regards to spectroscopy for identifying chemicals, as it turns out, both Raman and FTIR, I'm just going to show the Raman data, had really, really good accuracy and were highly accurate at identifying plastic particles from non-plastics and also identifying the specific types of plastic particles. It also turns out that novice labs can also get accurate results. So in other words, once you learn how to use the instrument, you can get good results. And the only real difference between the Raman and FTIR is that FTIR tends to, our data suggested that FTIR tended to do less well for the really, really small particles at 1 to 20 microns, which is consistent with FTIR having fundamental problems due to wavelength issues for particles that are basically that small. Raman tends to do a little bit better with regards to these smaller particles. So what are the things that are falling out from this study? Well, we now understand what these types of methods can and can't do. And this is stuff that's, that Scott has talked about and has posted onto the State Water Board website. It's currently being worked for in terms of peer-reviewed manuscripts for the journal Chemosphere as a special issue that is dedicated to the study. We also now have SOPs for these methods. These are now refined by the participating labs so that we achieve consensus on what works and what doesn't work and how. And the State Water Board is basically reviewing this. And for all methods, you need to be able to accredit laboratories so that, to show that they can have acceptable performance. We now understand what these, what the performance capabilities are. We know that what a good lab can achieve, and we're now working with eLab to develop the accreditation process for laboratories that would be doing this monitoring work. We have a website that is for all things microplastics in that we're doing, and that's all I've got. And I'd be happy to entertain questions. Thank you for your time.
Thank you, Charles. Thank you, Jeff.
All right, I'll go ahead and jump in and introduce our next speaker. Dr. Diana Lin is a senior environmental scientist in the Clean Water Group at San Francisco Estuary Institute. She studies microplastics and other contaminants of emerging concern and legacy contaminants in the San Francisco Bay surface water, sediment, fish, effluents, and stormwater. She and her wonderful team at SFEI just completed the first regional, comprehensive regional survey of microplastics in the San Francisco Bay. And she's going to talk about that and some of the monitoring methods development findings that they had. Take it away, Diana.
Great. Can you see my screen?
Yes.
Thanks. Thank you so much, Shelly. So I will be talking about the field sampling methods and lessons learned from our San Francisco Bay microplastic study. And as Shelly said, this was a huge team effort to conduct the study. So I wanted to thank my colleagues as well as Ezra Miller, who is attending as an attendee, who helped me put together this presentation. So we completed our study in 2019. This was the first comprehensive regional study of microplastics, and we partnered with 5 gyres as well as Chelsea Rockman at the University of Toronto to conduct the study. We had many study objectives. This included improving microplastic monitoring methods, both in the field and in the lab, so it's really exciting to hear all the new development and guidance on analytical methods. Identify baseline conditions of microplastic in the Bay, as well as share our findings and lessons learned widely. So I wanted to thank you for the opportunity to share our lessons learned with you all today. This project was a 3-year study that cost over $1 million. It was largely funded by the Gordon and Betty Moore Foundation, as well as additional supporting funding from these organizations. So I wanted to thank all of them for that. To address our many study objectives, our study design included sampling microplastics throughout the Bay, sampling Bay surface water, prey fish, sediment, bivalves, as well as sampling in the adjacent national marine sanctuaries. We also wanted to understand how microplastics were entering the bay in order to inform pollution prevention actions. And so we also sampled wastewater effluent and urban stormwater runoff, which in the Bay Area is largely untreated. Our full report, exciting 400 pages of reading, is available on our project website here. And we had many peer-reviewed publications that came out of this project, as well as additional ones that are in progress. This includes a polymer identification method, another one also by Zhu et al., synthesizing all the different matrices that we sampled in the Bay and did an analysis of that. General lessons learned from this project, which we've adapted that manuscript in this presentation. Some guidance on comparing different sampling methods and QA/QC guidance by Hung et al., and a more in-depth analysis of the stormwater results. So I urge you to look at these manuscripts for more details, and my goal in today's presentation is just to give you a high-level overview of our field sampling methods and lessons learned so that you could look for more details in these resources. So I wanted to start with some general considerations for the site selection, and Harry Allen mentioned that it's the importance of site selection in doing an environmental sampling. And particularly for microplastics, this is challenging because collecting and analyzing the samples is very expensive. So our study design was guided by our study objectives. And in order to understand baseline conditions in the Bay, we wanted to make sure we were sampling a good spatial distribution throughout the Bay, including sampling different, say, sub-embayments, sampling closer to microplastics pathways, such as effluent and stormwater discharges, as well as locations further away. We also sampled a reference site far away from the Bay Area so that we could look at compare our concentrations in the Bay with another location that is not so influenced by urban influences. And in terms of sampling different matrices, we wanted to co-locate our sediment sampling and our prey fish sampling to look to see if there were any correlations in the concentrations we observed. We also wanted to consider what were concentrations during dry weather and wet weather. And this was based on our understanding of other contaminants where, particularly in California, where we have a wet season, that can drive stormwater runoff, can take a lot of contaminants into the Bay. We got a lot of feedback as we were developing the study design. It was a long process to develop our study design, and even as we were implementing it, we were getting feedback. This included getting feedback from our Bay hydrodynamic modeler as to where we could optimize our sampling sites, talking to Chelsea Brockman, our analytical partner, on how best to collect the sample, what size sample. As Jeff Wagner talked earlier, it's important in terms of the sample size because sampling, analyzing the microplastic samples is really labor intensive, and so you want sufficient particles to get a representative sample, but you also don't want so much that you overwhelm your lab and they can't finish analyzing the samples. We also want to consider how we would analyze the data once we collected it, and we also talked to our QA officer to make sure we would have confidence in the data once we got our results. Of course, we could lay out our best laid plans, but once we're out there, there are logistical concerns. And so our recommendation is to have backup sites because there were times when we went out in the field and we had to adapt on the fly as to where we would collect our samples and what samples to collect. Another important part of the study design is the sampling method. So that's important, and that can change depending on your study objectives. And one important consideration is the sieve size and what size particles you're collecting. This is a graph where we graph the final effluent results reported from various studies. And this graph shows on a log scale the much higher concentrations reported for effluent depending on the sieve size you used. So this makes sense, as Scott talked about earlier with the power law for particle size distribution. As you sample smaller sizes, you'll see more particles. And then I wanted to just reference Hung et al. Who talks, and in this paper we talk further about how the sampling methods matter in the results that you get. QA/QC is really important for microplastic sampling. Microplastics are ubiquitous, and it is so easy to contaminate your sample. So it's really important to minimize and identify potential contamination sources. And even if you think you've addressed them all, it's still important to take field lengths so that if and when you see, you can try to quantify background contamination levels. We collected both field length and lab length so that we could differentiate potential contamination from the sample collection versus in the lab processing steps. We also collected field duplicates to see how variable the same sample would be if we collected it at 2 different times at the same location. So next, I'm going to just briefly describe our sampling methods for each of these matrices. I've just numbered them so that you could follow as I go through each of the matrices, and some brief lessons learned from our sampling. For surface water samples, we use both manta trawls and bulk grab samples. So the manta trawl is like pulling a plankton net across the water surface. So it works kind of like a manta ray skimming the water surface and picking up particles as it's skimming the water surface. This sampled through a large volume of water. The sieve was about 1/3 millimeter. So that's the size limit of the particles that we're capturing. In contrast with the 1 liter grab samples, we can sample a much smaller particles because we're not sieving them in the field, but it's a much smaller sample. What we learned from the sampling efforts is that the field duplicates can vary widely. In the manta trawl samples, the relative percent difference between duplicates ranged from 2% to 100%. And this just emphasized how ephemeral microplastics concentrations can be. For example, there were times where we could see that we were sampling through a foam line or a tidal front that had a bunch of debris and trash in it, and definitely a higher microplastic concentrations level. So our recommendation from this experience is to definitely pilot your testing method. It can inform what sample size you should collect. Like I said, you want to optimize you want to capture a representative sample, but at the same time not overwhelm your lab with the number of particles in each sample. It can also help identify contamination sources. As we were sampling, we identified potential contaminations in our field length. In this middle picture, you'll see there was a black mat on the boat, which was a contamination source. And then in our field length, we also saw bright orange distinctive fibers that were probably coming from our bright orange flotation devices. And so from that experience, I recommend collecting blanks from different steps to help identify where contamination might be coming from. And so if you don't get anything else from my presentation, my recommendation is to collect field lengths. I know field lengths are expensive because it's another sample to analyze, but it will give you more confidence in your results. Moving on to sediment. For our sediment sampling, we use a Van Veen brad and use the stainless steel scoop to sample the top 5 centimeters of sediment. This is what we do in the regional monitoring program, and it's more representative of current conditions as well as the layer sediment that is bioavailable. The way we collected our field lengths for sediment samples was to rinse the equipment with reagent water into our sampling equipment. We actually only collected 1 or 2 field lengths, which were actually quite high, So we think that they were a potential overestimate because most of the times when you're collecting your sample isn't touching all of the equipment. And the way to address that is just to collect more field blanks. Another important lesson learned is to record various units of measurement because different studies use different units. And so this is really easy to collect when you're in the field, but if you don't record it, then it's really hard to backtrack that information. So making sure you record wet weight, dry weight, volume, and approximate surface area that you're sampling from in the field. For tissue samples, there aren't any standard methods for collecting field blanks, so we didn't have a good field blank. We didn't have a field blank for tissue samples. For prey fish, we collected top smelt and anchovies, which are abundant in the bay and are important prey species in the bay food web. Our sampling method for the prey fish samples was to collect 10 individual fish at 6 different sites throughout the bay. And our method for the bivalves was slightly different, where at the locations, the tissue is a composite of 3 tissue samples. And so there are pros and cons with these different approaches. If you can get individual species, organism data, that's more useful, but then you have more samples to analyze. Some lessons learned is that we saw a large variation in the particle counts for individuals. So for example, for the fish, the distribution was skewed right, where most of the fish had very low particle counts, lower than the average, and there was a much smaller number of fish with much higher particle counts. And so it's really important to collect enough fish, different organisms in order to get a good representative sample. It's also important to be transparent of the fiber counts for comparison with other studies, because as we were trying to compare to other studies, we found that a lot of studies don't report fiber counts because it's so easily a contamination source, and therefore you need to be able to report microplastic concentrations with and without fiber counts. And we found that lab blanks were extra important for these tissue samples because the lab processing step is very long for digesting the tissue, and that is a potential contamination as your sample is sitting in the lab digesting. For wastewater effluent, we collected final effluent samples, and that was run through a stack of 2 sieves, 125 and 355 sieves. In order to collect a representative weekday sample, we collected a 24-hour composite on a weekday. And our study design was to sample at 4 facilities with secondary treatment and 4 facilities with advanced filtration methods, so in additional to the secondary treatment. And we wanted to be able to compare any differences in microplastic concentrations from facilities with these different treatment methods. Some lessons learned from this process is that the conditions at each facility can be very variable. There will be different concentrations in the sample. Your sampling location might be indoors at one site and outdoors at another site, and so the potential for contamination is very different. So it is helpful to have more field blanks again. Even with our 125 sieve size, it was very easy to get clogged over a 24-hour period. And I know this will be an even more challenge if you're sampling much smaller particle sizes. For stormwater runoff, we use a similar sieve size as the wastewater sample so that we could compare these 2 pathways. We used a 125 and 355 micrometer sieve, used an ISCO pump to sample our stormwater location. Our sampling approach is to take sips throughout the storm and then to take a depth integrated sample by tying the end of the ISCO pump to a pole that we could move up and down the water column. So the depth integration is really important so that we could extrapolate our concentrations to potential loads based on flows. This is actually more challenging than we expected. We think we might not have gotten the full depth integration, maybe undersampling the surface water and closer to the sediment bed. There were some sites where there are much higher concentrations, and so it would be helpful to be able to adjust the sample modeling based on what your expected concentration might be at the site. One of our big findings from this study was us identifying abundant black rubbery fragments that we later were able to link to tire wear. And these particles are actually really challenging to identify the polymer with FTIR and Raman, so they require other analytical methods to sample. And as we did further literature review afterwards, we realized that most tire wear particles are much smaller than the 125 micrometer sieves that we use. And so if you want to get a more representative sample, it would be better to use a smaller sieve size. So I just wanted to summarize my key takeaways from our field sampling effort. Again, do some pilot testing before you collect the samples because you might not know what to expect, and you can adjust your study design accordingly. You can adjust your protocols, refine your methods, refine the sample size that's being collected, and that can help you inform having to adjust your budget needs and time needed for analysis. Sampling may be necessary in terms of particle analysis. We found that when we had some samples that had way too many particles to analyze. And have a backup plan as you're collecting field samples, as you're analyzing samples, and you might meet budget constraints. As I mentioned at the beginning, you can see some of our full report as well as these peer-reviewed publications for additional information. Another recommendation is to have carefully designed field and laboratory templates to help to make sure you write down all the important field parameters when you're collecting the sample. We've shared the templates that we use on our project website, so you can go to that project website for all of our microplastic resources. We actually have a webinar that is recorded and posted on our website describing some guidance on study design, analysis, and reporting. Please let me know if you have any questions. This email goes to me as well as my microplastics team at SFEI.
Great, thank you, Diana. It looks like there's a number of questions in the Q&A box. Let's go ahead and have you answer one, and we'll move on and we'll come back to those if we have some time. The first question, were the particle sizes based on actual measurements or just a count of the particle captured in each sieve fraction? It seems like smaller particles could get caught in larger sieve sizes due to accumulated sample debris that are captured along with the microplastic particles.
Yeah, that's a great question. Is this about the graph that I showed?
I believe so.
Yeah, I think the limit is based on the sieve size and not just the particle size. And that's also true for our sampling as well. Like we use the 125 sieve, but yes, smaller particles can get caught in that, and you will get some counts of smaller particles, but your sampling isn't as representative because some of the smaller particles can often escape.
Great. Okay, let's go ahead and move on. And if we have a chance, we'll come back to those other questions. And you can review them as well, Diana, in the Q&A box.
So it's my pleasure to introduce the next speaker, which is Dr. Andrew Gray. And Dr. Gray is an assistant professor of watershed hydrology in the Department of Environmental Sciences at the University of California, Riverside. He completed his PhD in hydrologic sciences at the University of California, Davis, where he focused on the sediment dynamics of small mountainous rivers. At UCR, Dr. Gray has built a research group that investigates the processes controlling water and sediment transfer, with particular interests in wildfire impacts on sediment dynamics and water quality, coastal sedimentology and geomorphology, as well as trash and microplastics pollution. And as an aside, I've been working with Dr. Gray for the past few years as I did my PhD at UC Riverside, and I will hand it over to Dr. Gray.
Great, thanks a lot, Scott. And it's a real pleasure to be here and have a chance to chat with you all. And it was great to hear about SFEI's study just before mine, which is at a much more mature stage, and we've certainly been using the lessons learned from their study to guide the work that we're doing. Can everybody see my shared screen for my slides?
Looks great.
Great. So today I really just want to kind of focus in on microplastic fate and transport in watersheds and highlight some of the monitoring needs that we've identified and are trying to answer through our work to better support the kinds of data analysis and modeling that we want to do to better understand fate and transport. First, I'd like to thank all the students in my lab that are really in the trenches, in the channels, on the bridges, in the laboratory doing all of this work. They've been working very, very hard and continue to do so as we move towards the wet season, which always gets really crazy for us. So I wanted to start off with a note from Earth surface processes because that's my background. I'm a hydrologist and geomorphologist, and I really specialize in natural sediment transfer in watersheds and coastal oceans. And really, I came to microplastics with the perspective that they're really just another kind of particle that's moving through the Earth's system. And their closest natural analogs are the finer fractions of mineral and organic sediments that we find throughout watersheds in the Earth's system. And these sediments are commonly transported in suspension by rivers. And for that reason, we can use established approaches for analyzing and modeling and monitoring the source, fate, and transport of suspended sediment to then guide our approaches for microplastics. And so, as we think about microplastics in watersheds, why would we decide to monitor those microplastics in stream flow rather than, say, perhaps starting out with a much more distributed fashion across the watershed? There's a number of reasons for this. One of them is that, the efflux of microplastics from stream flow is the source or the boundary condition for the next receiving body downstream. A lot of this work on microplastics was really engendered by initial interests in microplastics contaminations in the marine environment. And then identification of rivers as a primary source has pushed even more of that work upstream and into the fluvial system. Also, by studying microplastics in the river, we're gaining understanding of potential impacts on that sampled reach and associated systems. And then we can also use that sampling and monitoring and then modeling within rivers to help us begin to start deciphering upstream controls related to the source, transport, and transformation of microplastics through the watershed. And so, as I mentioned, watershed microplastics are like natural fine sediments in many ways. We know and we suspect and largely know that they're dominated by non-point sources at this point, in part due to the efforts of SFEI and many others. Microplastics are everywhere. Of course, point sources are potentially very important because in some places you have much higher concentrations of microplastics that are being introduced to the environment. We also know that fundamentally the introduction of microplastics into the channelized system of a watershed, it's a transport limited phenomena when we're on the hill slope, when we're outside of the channel on these terrestrial surfaces. But once we get into the channel, this is primarily a supply limited phenomena, which means that most channelized flow can move most microplastics. Now there's many different ways in which microplastic sourcing and transport can differ systematically from natural sediments, such as, we expect shallow surface abundance on hill slopes or these terrestrial surfaces rather than, ubiquitous sediment supplies throughout soils and across the Earth system as we have for natural sediments in some cases. We also expect to find much more of them in urbanized systems, and this has been borne out through a lot of initial monitoring and also washing off of impervious surfaces, which tend to actually shut down the supply of natural mineral sediments. And there's other systematic differences in source mobilization and exhaustion thresholds. And also, when it comes to microplastics, we potentially have a better shot at utilizing the plastic particle characteristics to give us an idea of source than we may have for a lot of mineral and even organic natural sediments. So as we move into the channel itself and thinking about how microplastics are transported in rivers, as Dr. Lin mentioned, we're really dealing with particles that are moving through rivers, so they're not necessarily being transported in a fully mixed fashion throughout the flow field or in this two-dimensional representation down through the water column. This introduces complications for monitoring, and it also has important implications for transport. One of the important implications for transport is that it solidifies the idea that we're dealing with a supply limited phenomena in the channel. Most of these particles that are moving as suspended load with our natural analog the flow could potentially transport tremendous amounts more than we almost ever find in the channel. And that's also certainly the case for all the microplastics that we're dealing with under most flow conditions, rather than transport limited conditions where you're dealing with larger materials that are much more difficult for the flow to move and can only move during higher discharges. So this has implications for not only how microplastics move through rivers, but also how we can utilize samples collected from rivers to better understand the sources of microplastics in rivers as well. Another point to make in terms of downstream impacts on the next receiving body, in this case, sort of the ultimate sump, which is the world's oceans. When it comes to terrestrial sediments and their flux from the continents into the world oceans, that flux is really dominated by active margins. So areas where you have lots of uplift that's generating young mountain ranges, generating a lot of sediment supply. That's then that supply is then moderated by transfer through watersheds, and small mountainous watersheds then become the most effective supply and transfer agents because you have less of this internal storage in floodplains to hold that sediment back in part. Plastics, and this diagram from Meyer et al. 2021, this is focused on bulk plastics rather than just on microplastics, but as we know through particle size scaling, et cetera, that these patterns you would expect to be relatively similar. These are the results of in both of these cases for natural sediment and for plastics. This is a result of some monitoring, a tremendous amount of more monitoring for natural sediments than for plastics, but still monitoring that's then being utilized through empirical models to estimate these fluxes. With plastics, this empirical approach has tended to utilize the human population as the engine of supply, which you would expect. And then we have the watershed as a moderator for transfer, but also solid waste management as well. One thing I want to just point out briefly in this talk is that in both cases, we tend to monitor, when we are monitoring the flux of these materials through rivers, we tend to monitor upstream from tidal influences, from coastal marine influences in the rivers for reasons related to being able to accurately characterize flux. But there are potential, there is processing of these sediments within the coastal marine zone, within estuaries, and this is something that has been studied for a long time for natural sediments, but still remains a problem for closing down some of these budgets. And it's also a need for microplastics at this time as well. So when we think about monitoring in rivers, Dr. Lin already went over this in great detail, but the point that I want to really make is that we need dynamical or flux-based monitoring rather than ambient characterization. I'm not saying, and so by ambient characterization, I mean going in and dipsticking a stream, just measuring concentration or character in the absence of monitoring the flux of water through the river, the discharge of water. I'm not saying that we shouldn't do that at all, but we have a much greater need for monitoring microplastics concentrations when we're also monitoring discharge. One, because this allows us to estimate flux for that instant or time period of monitoring, but it also gives us the tools to start moving into empirical approaches for estimating and predicting flux, and then also moving upstream to consider the dynamics of the watershed supply and transport continuum. So as we start thinking about monitoring a little more, and I'll go through this quickly since this has already been covered, but really in keeping on with this comparison to natural sediments, microplastics have a much broader range of particle characteristics than natural sediment. We see a broad range of characteristics with natural sediments, particularly if you go across the mineral to organic spectrum. But with microplastics, we're talking about a wide range of sizes, a tremendously wide range of shapes, because we also have films and fibers that can be very prevalent, but also a wide range of densities, which leads to dealing with particles that fall within this suite of contaminants that are, some are strongly negatively buoyant and some are strongly positively buoyant, which is very different than what we find with natural sediments, which are mostly negatively buoyant. And so for this reason, when we start looking at where we find these microplastics within the flow field of the channel, within the cross-section of the channel, if you're thinking about it three-dimensionally, if we go through and plug in the boundaries of size of microplastics, of the full range of densities that we expect to find, and then the wide range of commonly encountered flow characteristics, because these are the major aspects that conspire to determine where we will find these particles in the water column in a river. If we go through and do this work, and this is work that was published by Wing Kauger from my group back in just this past year, what we find is that we would expect to see microplastics actually moving through the river under all different modes of transport that we've identified for microplastics. So that includes, in some cases, for some extremely low energetics in the flow field, bed load or even immobile microplastics in some cases, but settling suspended, which we commonly find with mineral suspended sediment, and then on into surface load, where we find most of the plastics at the surface, or rising suspended, where we have higher concentrations towards the surface for those low density plastics that have a positive buoyancy, as well as wash load, right? Particularly as we move into the finest fraction, where we have fully mixed and uniform concentrations across the flow field. So that means that really, if we do want to try to wrap our arms around the full diversity of microplastics and flows that are possible, that will require flow integrated sampling. But that's not always our goal, and that's something that we really need to think about as we move forward with monitoring microplastics. And if we're focusing on things, say, that we know we'll find in wash load through simple fluid mechanics calculations, then it makes sampling much, much, much easier. So as we move into what we can do with some of this monitoring, we have various watershed modeling approaches as we start to get at estimating abundance and flux and predicting it as well. So, a very commonly used approach in hydrology are these physics-based distributed hydrologic models. These are models that have very high data demands for appropriate calibration and validation in order to predict abundance and flux. But without full calibration and validation, they can be used to provide insights into system function, right? And I think that's really where these kinds of more sophisticated models can make are starting to make and will continue to make their initial contributions to the field, rather than by trying to use them to actually estimate and simulate abundances and flux in the systems that we're studying right now, because we just don't have enough monitoring. And I don't think that we will for a while, if ever. The other real benefit from these is that you can eventually use them to simulate unobserved phenomena if you have enough monitoring. The other approach is empirical, so it's using rating curves, which I'll go into in a second, or lumped or even semi-distributed models. These require less data. We can still derive some insights as you start moving into fully unobserved phenomena, they can be problematic and very limited. But I'll move forward with mostly focusing a little here on empirical models, because that's the initial approach that we're taking. So as we move into empirical microplastics flux dynamics, the starting point is really, again, flux-based monitoring, right? Where we're monitoring concentration. That's what you're seeing here on the y-axis. This is all log transformed, and discharge. This is a suspended sediment concentration rating curve in this bivariate space, which is the most common empirical approach that's used for taking monitoring data at a station on a river for suspended sediment, and then moving forward by utilizing primarily discharge monitoring alone in order to predict microplastics concentration and flux, right? And this is what we're starting to do with microplastics as well. It's the beginning of modeling, essentially, for fluvial particulate transport. And so there's the details that go into this, but essentially, because we're dealing with primarily a supply limited phenomena, you might ask, well, why is there such a large dependence, in this case, of suspended sediment concentration on discharge? That's because the same internal and external factors, so by internal, I mean watershed characteristics, and by external, I mean exogenous factors like climate and human forcings, et cetera. The same factors are conspiring to, in a lot of ways, influence the supply of water to the channel, that also influence the supply of sediment to the channel. And we expect that this is also largely the case with microplastics, although we think it will also systematically differ, and we're already seeing some evidence of that. And so for that reason, microplastic concentration then has a lot of residual dependence on discharge. Now, we can utilize that primary dependence in order to help us estimate flux, and think so that we can estimate what's happening when we're not monitoring at our station, and then also, what our fluxes are to the next downstream body. But we can also then go back and further interrogate these data to help us better move upstream and understand something about these forcing factors that are conspiring to introduce water and, in this case, microplastics to the channel. So if we look at suspended load transport in rivers, particularly as you move into the small mountainous rivers that are typical here in California, or draining a lot of California anyway, we see huge ranges in concentration. If you consider the entire range of discharge magnitudes that are sampled, it's very common to see 4 orders of magnitude of variability in suspended sediment concentration. And then even after we go through and fit a rating curve, which is our best first simple empirical step to being able to estimate concentration through discharge, we often will have some on the order of magnitude on either side of that fit of variability. And so, as we start to look into plastics, this is some work from Win Calgary again in my group that's in review currently. This is actually work in the Santa Ana River on fine macroplastics, so the above 5 millimeters, but not too much larger than that. These are count-based concentrations. Here again, we have discharge on the x-axis. Both are, log space. This preliminary analysis, if you go through and fit a more complex model to this, you can make it fit pretty well, and it looks like it's relatively tight. But if you're going to try to fit a power law relationship, which is this log linear relationship we see here, you see a lot of variability as well. And so this is further caution against ambient monitoring, because we know we have so much variability across the discharge domain, but even within any given discharge magnitude as well. We expect to find that, and we're starting to find it. Other complications include hysteretic behaviors. So this is path dependence, which can also be further generalized to temporal dependence. Many of you are really familiar with this at the seasonal scale, first flush dynamics, especially here in these dry summer Mediterranean climatic regimes that we experience in California. you have this entire dry period where very little, where no rainfall runoff and fluvial activity is happening. And so oftentimes during the first flush, the first sort of effective rainfall runoff event of the season, we see very, very large concentrations in many fluvial constituents, particularly particulates. We've expected to see that for microplastics. With some of our initial findings here for coarse macroplastics, we're not. If you look at the dates on some of these, we see actually higher discharge magnitudes earlier in the season with lower concentrations. We see some initial evidence for other scales of hysteretic behavior, such as rainfall runoff event, hysteresis. This is the difference in concentration as you move up and then down the hydrograph. So same discharge on either side of the hydrograph, but very different concentrations. We don't have enough monitoring yet to really document that, but we see it sort of pasted across the larger scale. And we expect to find more of it for a number of reasons that I won't go into now. But the takeaway from all of this is that with these kinds of additional dependencies on time and path, it increases our monitoring demands to adequately characterize dynamics, even for these simple empirical approaches. So if we move beyond the simplest of empirical methods and move into the next step of complexity is lumped empirical modeling. That this relies on watershed composition, hydrologic regime, et cetera. These are the kinds of models that are being used to estimate global fluxes of plastics, and people are starting to use this for microplastics as well. You can then use this to predict the abundance of flux in unmonitored watersheds. But it requires that you go through and monitor many watersheds that represent a wide range of compositions and hydrologic regimes. And then you can utilize that to develop some kind of empirical model. And that is one of the ways forward, so that we don't have to go through and monitor every single watershed. And we already have inklings and indications of this, that we'll find much more microplastics, say in watersheds that are highly urbanized versus or highly impacted by agriculture versus undeveloped. But we need more work so that we can actually move forward with this approach. And that's one of our goals in my lab as well. Beyond that is, to better inform lumped approaches or even move into semi-distributed empirical approaches that will require finer scale subbasin analysis and even source evaluations. People are, of course, working on this already, and this will help as we move towards potentially these more complicated models. It involves interrogating major point sources, even looking at hill slope to subbasin specific studies, and also trying to close in on additional transport processes that are introducing microplastics to our watersheds or channels or aquatic systems, including aeolian transport processes, which may be quite important. So to wrap this up, and to really focus on the needs and recommendations for microplastics monitoring, we need to do more monitoring in estuaries, both water and sediment, to better understand the processing of microplastics as they move through this transitional zone between the fluvial and coastal marine environment. We need to focus on flux-based monitoring of microplastics in stream flow, and we should focus on storm flow. And I didn't go into this as much, but as we think about effective discharge of sediment in the kinds of systems that we're dealing with for natural sediment, effective discharge, which are the ranges of discharge that transport most of the sediment, is mostly higher magnitude, lower incidence, so lower frequency discharges. We expect that won't exactly be the case for microplastics, but that storm flow itself will be much more important than lower flows, base flows, or some of these urban drool or wastewater return flows. We need further investigation of microplastics transport dynamics in stream flow in coastal marine environments, which means directly investigating differences in concentration and character across the water column. And that can then go on to help inform local impact assessment and flux predictions as well. And we need more information on what classes of microplastics to prioritize, so that as we move beyond academic research groups such as my own and others that really specialize in sediment monitoring in rivers, that we can go and advance techniques for monitoring that are easier to implement less effort and equipment intensive, and can hopefully piggyback off of infrastructure that's already in place in a lot of these rivers for monitoring other constituents. And then we really need, moving beyond that, and well, I guess that's the next point, right? That we need representative streamflow monitoring. And if we can narrow down some of this diverse suite of particles to the highest priority microplastics, it'll help us to do that more effectively. And then finally, direct and indirect monitoring of some of these potentially important point and non-point sources and additional transport processes is very important, and I think will be more important as we move into the future, specifically on wastewater treatment plant biosolids, land use specific runoff, even in-channel storage dynamics, which I didn't touch on much today, but we're seeing evidence that in-channel processing in these, particularly in our kind of ephemeral systems, can be really important. And then aeolian transport and depositional processes. So I've kind of set a lot of things up and then didn't talk a lot about the studies that we have underway because they're underway, and I don't really want to talk about our preliminary data yet. I've talked about some of our preliminary work in the Santa Ana River, but we have ongoing projects in San Pedro Bay and the major watersheds draining into San Pedro Bay as funded by the NOAA Marine Debris Program in partnership with our co-PIs at SCWRP and in partnership with Orange County Sanitation District. We also have ongoing work in Newport Bay as funded by the State Water Board and in collaboration with Region 8 and with Orange County Environmental Resources. And then we're beginning pilot studies in collaboration with LA County Public Works to really investigate the opportunity to utilize some of their standing infrastructure to sample microplastics in ways that are much more efficient than our kind of full boogie research-based approaches. And so with that, I'll close it up, and I'm ready for questions if I have any time left. Thank you.
Thank you, Andy. So I think in the interest of time, we're going to go to the next speaker, although I do want to entertain just one of the questions, and the rest, I think, Andy, if you could just fill those in the chat. So someone asked, you mentioned the high variability of the stream samples. We've seen similar variability when comparing samples using low volume, i.e. below 1,000 liters. Would it be possible to test whether volume is affecting the amount of sampling error, perhaps by sieving in situ, such as was done during the SFEI wastewater sample collection?
Yeah, that's an additional great concern, right? Because for natural sediment, oftentimes we're also introducing a pretty large amount of uncertainty through our sampling techniques. So, how certain are we that our sample that we've extracted from a flow is representative of sediment moving across the flow field? And this becomes probably even more problematic for microplastics because of the lower concentrations that we're dealing with and a broader range of particle characteristics. And so it makes all this it introduces even more uncertainty, and we would expect to see even broader ranges of concentrations. Yeah. So it's all the more reason to do some replicate work.
Fantastic. Thank you, Andy.
Great.
I'm going to pass it back over to Shelly.
All right. So I'm going to introduce our next speaker. It's Dr. Susanne Brander. She's an assistant professor at Oregon State University. She's an ecotoxicologist whose research integrates responses of aquatic organisms to endocrine disrupting compounds and other environmental stressors such as microplastics across the biological hierarchy. So it is a pleasure to have you, Dr. Brander, and I'll turn it over to you so you can talk about risk assessment.
Great. Thank you so much, Shelly, and thank you for having me today. Like was explained, I'm going to be talking about microplastics risk assessment. I'll be going over several processes underway on the West Coast, 2 that are underway and now near completion in the state of California, and a 3rd that is a collaboration between OSU, Western Washington University, and some of the great work being done at the San Francisco Estuary Institute, all to assess the risk of microplastics in ambient waters. And the work I'll be talking about today is part of several very large collaborations and is the result of the contributions of many. So I just want to acknowledge that before I begin. So first, I'll give a brief introduction on the toxicity of microplastics and why we're concerned about risk. I'll talk briefly about the California Ocean Science Trust report that came out earlier in the year in the spring. We'll talk mostly about the California Microplast Health Effects Working Group that was convened by the Southern Coastal California Water Research Project and the California Water Boards, and then talk a bit specifically about Bayesian modeling approaches that are currently underway between my research group that I co-lead with Dr. Stacy Harper at OSU, which is the Pacific Northwest Consortium in Plastics, and some work we're doing in collaboration with SFEI. So what responses are seen to microplastic exposures? And others in this webinar today have referred to these responses here and there, but we see a myriad of different effects in aquatic organisms. We, as a large research community, are beginning to converge on some commonalities here. 2 in particular that I'll focus on today are impacts on growth, likely caused by food dilution or false satiation when an animal ingests a microplastic, as well as oxidative stress, which may occur due to translocation. It's also important to acknowledge that some studies see no effect, and often impacts are only observed at high concentrations. But we are beginning to see these common responses across different taxonomic groups, as well as across biological scales. What do we know about toxicity? To briefly discuss this, we know that size and shape influence toxicity in laboratory studies, and that evidence shows that fibers may be more toxic than other plastic shapes or morphologies, and that the smaller the microplastic is, and even getting down into the nanoplastic size range, so below a micron in size, those smaller plastics appear to be more likely to cause oxidative stress. Upon exposure to microplastics, cells can produce reactive oxygen species, and then these ROS can cause structural damage, and some of this may be due to translocation. And again, like I said in the last slide, food dilution and translocation appear to be major drivers of toxicity in aquatic organisms. But of course, we all know that we can see plenty of responses and indications of toxicity in laboratory exposures, but those responses don't necessarily reflect what's happening in the wild unless that exposure is in the lab is relatively similar to what's happening in the field. And so it's really only when that hazard and exposure overlap that you're able to accurately estimate risk. And that's the aim of several of these efforts that I'll talk about today. So 2 state-level actions in the state of California were the Ocean Science Trust Science Advisory Panel. And this began right at the start of the pandemic in spring of 2020. And this was a qualitative risk prioritization framework that was put together specifically for marine ecosystems in the state of California. Report came out earlier this year, really relied on the precautionary principle, source reduction. So limiting the inputs of plastics into the aquatic environment was emphasized. This was all convened by the Ocean Protection Council and grouped scientists from the US and Canada to work on this issue. And then the Microplastics Health Effects Workshop, which is a much larger effort, was convened by SCWRP and the water boards, and this involved experts from Europe and North America. It resulted in the creation of the TOMEX database, so data from 167 different peer-reviewed papers, and I'll talk about the species sensitivity analyses that were done from those data. And then the goal of this was threshold derivation for ambient water and for drinking water, although today I'm just going to focus on ambient water in the interest of time. And so in response to State Bill 1263, which Dr. Coffin referred to in his presentation, the group convened by the Ocean Science Trust came up with a risk characterization and ranking process. And again, this is qualitative, and we were a bit limited because the focus was primarily on the state of California and on species of concern in the state of California, for which right now there's really pretty limited data. But in the meantime, we came up with an approach that includes some key factors to be considered, such as particle morphology, polymer types, priority species or endpoints, fate and transport, and sources. And the outcome of this is that one can use this framework to characterize and rank risk based on the amount of microplastic that is estimated to be loading into the environment and the risk of ingestion or internalization by priority species. And then, of course, because we're limited in those data for California species, surrogate species can also be used. So it basically prioritizes source reduction strategies and allows consideration of risks that might warrant state action or potential mitigation. For the Microplastics Health Effects Working Group, which was convened again by SCWRP and the water boards, this involved scientists from North America and Europe, and tasks for the group were to agree on a management framework for microplastic assessment in ambient waters, derive a method to produce thresholds, species sensitivity distributions were used, and I'll talk a bit about this, and all of the studies considered holistic analyses underwent very strict quality assurance and quality criteria. Be familiar with, but have been used for decades. They're a probabilistic method to derive thresholds for toxicity of a variety of different environmental stressors, different groups of contaminants. Often values for algae, crustaceans, mollusks across a number of different taxonomic groups are used, usually relies on laboratory testing. And the goal here is to calculate an estimated hazardous concentration for a large majority of species. Usually the cutoff is at an HC5 or an HC10. So you're protecting either 95 or 90% of species in that SSD. For the Microplastics Health Effects Group, separate sets of thresholds were derived by aligning data based on two primary mechanisms that I talked about a little bit earlier. The first was food dilution, which can result in limited growth and other impacts like limited reproduction, and then tissue translocation, which is thought to drive these oxidative stress responses that are being seen in many different types of organisms exposed to micro and nanoplastics. So I certainly don't expect you to take all of this in. There's a lot going on this slide, but these are the 4 species sensitivity distributions that were derived from this ambient water working group. The ones up top were used for food dilution, and after QA/QC was performed, we ended up with studies that represented 15 species across 6 different taxonomic groups. And then at the bottom, these are the species sensitivity distributions used for tissue translocation. And here, after QA/QC, we ended up with 16 species and across 6 taxonomic groups. So enough of a representation across taxonomic groups and studies to derive accurate SSDs. And another important thing to mention is that, of course, most of the laboratory studies that were in the database being used for this effort are monodisperse, or people were exposing animals to one type and one size of microplastic or nanoplastic. And so these monodisperse concentrations were rescaled to a default polydisperse size range of 1 to 5,000 microns. And then if the particles were larger than the organism in question's mouth opening or were too large to translocate, those particles were excluded. So the ultimate goal here was to come up with a decision framework that could be used by the state to evaluate microplastics as a stressor. The framework you're seeing here is similar to past frameworks that have been used for other chemicals and other stressors. And the development of this management framework really requires generation of analysis that informs several thresholds, and each of those thresholds is linked to some sort of management action. And the lower thresholds are meant to be protective of effects on aquatic organisms, and so exceedance would require something more minor like investigative monitoring. And then as you get to those higher levels of concern, that's where it would lead to more costly management actions such as mitigation. So earlier decisions are inexpensive and reversible, and later steps have a larger cost. And all of this work is currently in prep in a manuscript being led by Dr. Alvina Mahinto from SCWRP. These are the species sensitivity distribution parameters and data filters used to derive thresholds for the tiered framework shown in that previous slide. So again, HC is hazardous concentration. The 5 represents 5% of species, and so the HC5 would be protective of the other 95%. You need to make that cutoff somewhere, especially since when you have a species sensitivity distribution, you have a lot of variability in that lower side, those lower endpoints. And you can see as one moves up the threshold, the criteria need to exceed that threshold are increasing. So we go from HC5 to HC10. We go from including molecular to population level endpoints to just including organism and population level endpoints as you get higher up those thresholds. For food dilution, we are estimating thresholds for food dilution as well as for translocation and oxidative stress caused by translocation. For food dilution, there's a big difference in threshold calculations depending upon whether one includes all of the endpoints, only fitness-related endpoints, so things that are going to affect an organism's ability to survive and reproduce, or if you limit it just to mortality, those concentrations do get much higher. And just to put this into perspective, 0.2 particles per liter for that threshold 1 seems like a very low threshold, but if you're thinking about this from the perspective of a smaller organism like a daphnid or a larval fish used for aquatic toxicity testing, that's roughly 200 particles per milliliter, which isn't as low as it sounds when you're putting it in particles per liter. Similarly for translocation, and remember this is for particles that are 83 microns in size or smaller, and that's the estimate of how small a particle needs to be in order to translocate within an aquatic organism. Threshold values also increase here depending upon whether all endpoints are considered versus just fitness related or only for mortality. And really in microplastics studies and lab studies, mortality is fairly rare. Usually you're seeing sublethal responses. At present, reported concentrations for both the translocation and the food dilution SSDs are lower than these derived thresholds, but plastic production is predicted to increase exponentially by 2050, and thus if we continue business as usual, as predicted by several recent publications, this will likely lead to higher environmental microplastic concentrations. So it's good to be a bit precautionary here and to think out to what future exposures might look like for aquatic organisms. So the vast majority of studies in the TOMEX database, and again, there are 167 studies total, and this was great work done by Dr. Leah Thornton-Hampton at SCWRP and Dr. Scott Coffin, pulling together all of these data that are being used both for generation of the ambient thresholds as well as generation for screening levels for drinking water, which I'm not going to discuss today. But the vast majority of the studies in this TOMEX database are done on spheres and fragments. Spheres in particular are not necessarily representative of what is detected in biota. As someone who goes out and samples fish and other organisms, I can definitely report this from experience, and you heard this from Dr. Lin at SFEI. And as you can see here, there are only 5 studies currently in the TOMEX database on fibers, which is what we really mostly find. And the number of studies in fibers is expected to increase as more data are added. And I show this to say that it's important to remember that the main goal of this work was to build a framework for the assessment of risk, and those thresholds will likely become more accurate and reliable as additional data on more realistic plastic types like fibers are included. And so just to hit this point home, thresholds may change as additional data are added because we know that fibers, both synthetic and natural, but of anthropogenic origin, are ubiquitous in water, sediment, and biota globally. And more studies on fibers have already come out since the database was completed, and are going to be added as they are published, and there are many more in progress. So moving past the microplastics health effects group and getting into some work that is specific to the Pacific Northwest Consortium on Plastics, which I co-lead with Dr. Stacy Harper at Oregon State University. And we also have Dr. Wayne Landis from Western Washington University, who is a PI on this grant. And all of this work is funded by the National Science Foundation Growing Convergence Research Initiative. And Dr. Wayne Landis's group, and specifically his current graduate student, Emma Sharp, focus on deriving risk assessments using a Bayesian network approach. So what is a Bayesian network? Well, I'm showing kind of a simplified version of that here. It's graphically depicted here is a web of nodes that link cause and effect relationships using conditional probability. And it's a way to describe interactions and to generate the probability outcome, or more often outcomes, multiple outcomes. So it allows for a lot of complexity. Another benefit of this is it's habitat specific. And so you're seeing the map here of San Francisco Bay microplastic risk regions. And the Bayesian modeling approach allows you to calculate risk for specific risk regions that are including characteristics of habitat, potentially land use that's specific to that area. And so it's a different type of approach from the species sensitivity distributions that were derived, in that the SSDs are a bit more generalized, where this can be specifically sort of targeted to a particular region. And in this case, because of SFEI's really groundbreaking study, there is a lot of great data to be used in order to inform a Bayesian modeling approach. And so Dr. Landis's lab and the rest of our group have been collaborating with SFEI, basically combining data from the lab with great data from the field. And just as a representation for one of these nodes, you're seeing one of the nodes in this network could be a laboratory study, generating data on growth or swimming behavior or mortality, for example, in a representative species. So, try not to spend too much longer on this since we're a little bit short on time. But what I'm showing here, and this is a lot to digest, is a Bayesian network for tire wear. And this is a preliminary risk assessment framework. And basically what you're seeing here is that everything starting from sources all the way out to impacts is included in this modeling framework. And under impacts are species of concern that can be impacted by a combination of factors. And again, this is work being led by Emma Sharp, pictured at the bottom there. She's a graduate student at Western Washington University. So low, medium, and high risks are based on the percentage of endpoint species of concern on the far right that are impacted by a combination of factors, in this case including tire wear particles, but also including other considerations like land use, transportation, ecological factors, and even whether it's the rainy versus dry season, for example. How does this all work? Well, each node can have up to 4 different states in this particular example, and then the percent of values in each discrete bin is predicted by the model. And so this is a Bayesian net example that's kind of hot off the presses, and this is work that's underway right now. And you can see we have the San Francisco Bay microplastic risk regions over on the left here. In the middle is data that is mainly being generated by the San Francisco Estuary Institute. So this is tire wear particle field data that has been collected from stormwater, sediment, effluent, and in surface water. And then over here on the right, we have lab data that is mainly being generated at Oregon State University by my lab and the Harper lab on a variety of different aquatic organisms. We also have some work being done out at the Hatfield Marine Science Center by the Langdon Lab on larval oysters, and so that work will eventually be included as well. And so as we select different inputs in one node, the distributions change in the rest of the model, and there are a lot of benefits to this. For example, you can have the model be reliant only on one of those risk regions. And so here, you're saying that 100% of this model is reliant on the San Francisco Bay region. And then you can also work backwards by changing the nodes at the end. For example, you could calculate what input would cause, say, a 20% increase in daphnid mortality. And then the ultimate goal is to use some of these surrogate species and data that we're getting from the University of California, Davis on internalization of microplastics in salmonid species to predict out to species of concern like Chinook salmon, which we know are affected by tire wear leachate. A study from Washington State was published earlier in the year on that, on coho and Chinook. So to sum this up and to make sure I give our last speaker enough time, the Ocean Science Trust Report has been finalized, is now public, is making its way to legislators in California at this time. Publications from the Microplastic Health Effects Working Group will be out in the coming year. And research recommendations from these efforts will certainly lead to the collection of data that better supports further risk assessment for microplastics. And from our group, the Bayesian model for tire wear particles is estimated to be completed soon. And then we will add in assessment of other particle types. There's testing with fibers, for example, and other plastic fragment types underway in labs at OSU at the moment. And I'll sum it up there, and thank you very much for your time. If you'd like more information on our work, you can check out our website, and we're also on social media. And just thank you very much to all of the students and to my postdoc, Dr. Samreen Siddiqui, who we couldn't do any of this work without.
Thank you, Suzanne.
It's my pleasure to introduce our last speaker. Last but not least, we have Dr. Terri Slifko. And Terri Slifko has devoted her career to the public sector in the field of water quality. Currently, she oversees sample collection, analytical chemistry, chemistry research, data evaluation, reporting, and other water quality support activities at Metropolitan's 6 water quality laboratories. Metropolitan is a consortium of 26 cities and water districts that provide drinking water to nearly 19 million people over a service area of 5,200 square miles, encompassing parts of Los Angeles, Orange, San Diego, Riverside, San Bernardino, and Ventura counties. Very pleased to have you here, Terri.
Thanks so much, Scott, and everybody. It's what a great meeting so far. I've learned so much, and I'm going to shift gears quite a bit and talk a little bit about drinking water, maybe a lot about drinking water. So I appreciate this opportunity to participate in this project. Metropolitan was one of the 40 laboratories that participated in the inter-laboratory method evaluation study. Our focus was on drinking water. And of course, we are motivated by SB 1422. So I'm going to talk a little bit about the next step of the inter-lab calibration study and developing a tier 1 monitoring program and how to do that. And so a little bit of background first. Since April 28th, when the Microplastics Method Evaluation Project reported out, potential new paradigm was introduced. That paradigm recognized limitations of the microplastics monitoring methods that were evaluated, and it recommended a tiered monitoring strategy. We recognized that some of the methods had strengths and limitations, and that this tiered strategy would answer slightly different questions on a tiered level of approach. And we really supported this because it helped us to get a little bit more time to develop methods that there were a lot of new methods that came on board since the project started. And so onboarding new methods that maybe work better than the ones that were evaluated or work better for us. So we recognized early that the methods had, some limitations in terms of timing and practicality, particularly for a routine monitoring program because of the high cost and uncertainty of those methods. So this Tier 1 monitoring group was formed, and it was formed to establish exactly how to proceed using inexpensive methods that we could use as surrogates or indicators that we already use in drinking water treatment plant monitoring. We're highly regulated, and we try to optimize our treatment processes as much as possible to make sure that we're meeting compliance requirements and operational goals 100% of the time. So I'm going to talk a little bit about that. I would like to recognize the Tier 1 group. This was a breakout group that met weekly, sometimes twice a week, to review the information that we had, to understand what options were available to us. And we really tried to get the Tier 1 monitoring plan completed by the end of by our deadline, so that it could be brought forward to the, that the state could consider our recommendations. But we couldn't, we didn't have enough information. And we realized during the vetting process that none of the methods that we wanted to use were ready at all. We didn't know, say, if you have 100,000 particles that are making it through your treatment train at a drinking water treatment plant, or even in your source water, what portion of those particles always, or most of the time with a boundary around it, could be microplastic. And so we realized we needed to conduct research and evaluate the correlations between online monitoring tools, real-time monitoring tools, things that we already use in a treatment plant that could give us this idea. So anyway, I just want to say thanks to all these folks here. We only had a couple of utility representations in this work group. And so, part of it was really trying to explain the limitations of monitoring in a drinking water regulatory environment. So it was fun, and I met a lot of new friends, and I really appreciated being able to work with them. So let's see if I missed anything here. Okay, so the goal of our group, this Tier 1 group, was to identify possible screening methods for for monitoring microplastics in drinking water. So not actually monitoring the microplastics, but an indicator of them. And that was our goal. And so again, we couldn't identify one single thing. There were no studies that were properly designed that gave us the data to support any good recommendations at that time, and or the total particle portion that were plastic, and we didn't have any correlation data. So we did identify some potential methods that we could use, and what was most important is we developed a process and recommendations and justifications for that process. So when we started the study, there were already a few studies that were published in the world that looked at drinking water treatment processes. Since then, several studies have been published. In 2019, the World Health Organization, WHO, reported that of 50 drinking water studies, only 9 of those studies were they were really reliable. The other studies didn't have the right kind of data, or they had contamination. But even those 9 studies out of 50, all of those studies had contamination issues in the blanks that they ran alongside of their methods. None of the methods were standardized, and so those studies were, initial efforts to try to understand if microplastics are in drinking water, and this was, throughout the world. So there are microplastics in drinking water, and in those studies reported between 0 to 100,000 particles per liter. The drinking water that was defined as bottled water, groundwater, and surface water, and groundwater under the direct influence of surface water. So it was a very wide range. Many different types of methods were used and many different ranges. However, let's see. I did mention that the QA/QC, all the data for their QA/QC for all those studies had, had microplastics in the laboratory control blank samples. So the slide that I'm presenting here was published in 2020, and they reported microplastics over 1 micron in drinking water in the Czech Republic. They looked at 1 river and 2 drinking water treatment plants that were really different from each other. And I like this study because they did a pretty stringent QA/QC here. They did find plastic particles in their blanks, but they did report that. And I really appreciate it when people, when researchers report, oh, hey, yeah, we had blank contamination. And then you can understand, okay, so that's the range and this is the error. So this upstream drinking water treatment plant was a very simple, conventional sand filtration and disinfection. And the other plant downstream was a more complex, advanced treatment process. And so understanding that's probably a few people on this call that aren't familiar with drinking water treatment plant processes. You get your source water, which in this case is river water. You add coagulants, and then you flocculate the water, settle the particles out. So the coagulants help to make things sticky, and then you settle out those coagulants. Then you filter whatever supernatant, after the coagulation process. And after settling, you filter that through, in this case, a deep bed sand filter. And this is a multi-matrix filter system. Deep bed sand filters are generally really good at removing small particles and large particles. Then it was treated by ozone and GAC, granular activated carbon, which helps to reduce nutrients. And then the water is served after. And I don't believe there was any chlorine used in this system. In Europe, that's very common. So most of the particles here were greater than 70% were fibers and fragments, smaller than 10 microns. The most frequently occurring materials were cellulose acetate, polyethylene, tetraphthalate, polyvinyl chloride, polyethylene, and polypropylene. This study is just the same study. It shows during the different stages of the treatment process how the concentrations of the particles sizes were reduced through the treatment process. And it was reduced in both plants. There were much less particles in the upstream plant, and they had less removal. Downstream plant had better removal. Upstream plant had less particles to start with, but they also had a sand filter, which is not as generally efficient as the deep bed sand filters are. This is a different type of treatment process. I'm not going to go into the details of this. I just want to mention that this study showed the same thing. drinking water treatment removed the microplastic particles and by over 85%. And this had some really interesting data, and it was recently published. And another study showed the same thing over the drinking water treatment process. From raw water to effluent, the microplastics were removed. And in this case, the particles, this was a GAC again. There were, I'm not going to get into details because I think we're running out of time. I thought I wrote down the type of plastic, but if you guys are curious about that, I'll let you know. I'm interested because I'm trying to figure out what type of instrument to buy to look at different types of plastics and what resolution and what library database I need. So this is, something I need to get ready for. So as you can see, these and other studies that have been completed since 2019 and the studies that were reported in the WHO report corroborated the findings from the WHO report that that microplastics are removed by drinking water treatment processes. And so one of the things that was struck me, and this is the question that I get from management that might read this report. And so other water agencies are going to say the same thing. Well, this World Health Organization, a group of international experts, said routine monitoring of drinking water is not recommended at this time, and there's no evidence to indicate a human health risk. So why are we doing this? You guys know more than anybody else probably in the whole world why we're doing this. So I'm not going to go into details, but the key thing is that water systems can be optimized. They can be upgraded if they need to be. There's other things that they're already designed to remove and incidentally remove microplastics at the same time of different size ranges. The WHO also said that, and these are recommendations, targeted and well-designed and quality-controlled investigative studies to better understand the sources and occurrence of microplastics in fresh water and drinking water, the efficacy of the different treatment processes and combinations of processes is needed. And also to understand more about the fate of waste streams in the environment. So that brings us to Tier 1 study. So if we know that drinking water treatment works well, what is the point of this conversation? Up to now, most of the world didn't look for microplastics in drinking water. And until now, there was no standardized and validated methods to give us this apples to apples comparison of removal efficacy. Different treatment plants have different source waters and supplies, and every single one of them has a different treatment process. They're not a cookie cutter thing. So everything's going to look a little bit different. And each of these different sampling protocols at a water agency are going to look a little bit different. Same like wastewater, right? So same thing, the source, wherever that comes from, impacts this, collection system in a wastewater agency. So the SCWRP Interlab Method Evaluation Study was able to standardize the microplastics methods for monitoring, but they also revealed several other critical and key issues. All the methods are time consuming and takes a great deal of technical expertise and cost, because the time is money and so are the instruments. And so it's really expensive. I'm going to beg for an instrument, and it's going to cost anywhere between $100,000 and $400,000, depending on what I'm going to do. So our most water agencies don't have funding to purchase such an instrument or the capabilities in their analytical labs. And a lot of these public agencies send their samples out to contract labs because they don't have the capabilities for complex analysis in their laboratories. So microplastics analysis takes a long time, and it's not practical for a routine monitoring program, but a Tier 1 is, and this is why. let's see. If you take the routine turbidity, TOC, particle count, DOC, or a handful of other things that you're analyzing, sometimes every 2 hours, and you optimize your treatment process, the question is then, is that result going to correlate with the removal efficacy of microplastics? So first we got to find out if they're there, and if they're high enough concentrations that we can draw a conclusion with this. So the purpose of this Tier 1 study is to plan a study to identify and correlate a good set of microplastic particle screening and removal indicators and surrogates to use for the Tier 1 monitoring. So right now it's an informal group. Today it's an informal group. You guys are all potential group members. And so I feel that it's time. And so I'd like to put together our heads and start thinking about how we're going to plan the next phase. How can we test, a small set of representative drinking water treatment plants in California and possibly Nevada to evaluate this, where we can compare plastics and other parameters, these surrogates. And so the purpose in the forum then therefore would be to develop a project scope of work. So the goals would be to establish the working group, to design and facilitate the project, test and rank the efficacy of existing surrogate monitoring tools. Then we could establish the tiered monitoring approach using the refined surrogates. So we'll find out what works best and whether we need more than one, and develop an indicator toolbox, if you will, and then use the tiered monitoring framework later to monitor and track the diurnal and seasonal fluctuations and trends that happen with rainfall and dry weather events and under different operational conditions. It would be really complex for Metropolitan because we have two main water supplies, and so different times of the year, we may be blending a certain percentage of one supply, which is very much different than the other supply, and sometimes it'll be all this supply and all that supply. So it's going to be tricky for us, but I'm hoping that we get a chance to do this. And I need you guys to help. Okay, so what we want is this tiered monitoring framework, what it's going to look like. We're going to develop a screening method. We're going to see if we can apply that for routine monitoring. And if there's a problem, then we go into bulk quantification, possibly by GC pyrolysis or something like that, something less time consuming. And if we do see something there, then we go into the time consuming methods. We characterize the particles that we see, we count them, and we find out where they're coming from. And then we develop source control to take care of the problem. Okay, so that's the rationale for Tier 1. And these are some of the items that we came up with in our Tier 1 working group. Some are more ready than the others. So there are a number of other recommendations and things that we pulled together. So when we do have a kickoff meeting of this Tier 1 working group, we'll pull all this information together. We'll have an opportunity to talk about it. But we did have like vendors going, oh, I have the perfect monitoring tool for you. And well, I've never used it before in a real system, but it's a great idea. So we have a lot of things to vet and test and try. We also need possibly somebody to pull the group together and keep it going. Money, probably, the methods are expensive and so forth. So we have some options here that would be fantastic to test. Some are, more ready than others. But the point is that we wait until Tier 2 to do the heavy lifting in terms of microplastics analysis. Okay, so
hey, Terri,
yeah.
I'm sorry, I'm going to have to cut you off. We have just 2 minutes left in the webinar. So.
Oh, okay. All right. So let me just tell you, there's a little bit of the work being done elsewhere, and then this is a monitoring framework. I'll send Scott the slides, so if you guys want them, you can have a look. And that is really all I had. Scott, I hope I did a good enough pitch for you.
That was great. Thank you, Terri.
You're welcome.
Sorry to cut you off. This has been a fantastic webinar. Thank you all for attending and all of the fantastic panelists that we had. Big round of applause for everyone. So this is just the beginning. And as you saw, we've got a lot of work that we can accomplish through this working group. And I'm looking forward to hosting our next meeting. And I'll hand it over to Shelly for some concluding thoughts regarding that.
Yeah, so I mentioned the trash monitoring workgroup, and we usually have quarterly meetings. I try to get some different types of speakers in from all over the world to talk about what they're working on. We have 3 subcommittees. We have encampments, data standardization, management, coordination, and citizen science. If you're interested in participating in any one of those, please give me a shout out, and we can get you hooked up with those folks. And what we're looking to do is create a microplastics subcommittee. And some of the goals for that subcommittee is basically bring together all people working on microplastics to look at creating a standardized method playbook, perhaps sharing information on current research, also informing those policymakers and decision makers, and most importantly, we can't all work for free, looking for opportunities to collaborate and get some funding. So make sure to contact myself or Scott if you're interested in being part of the subcommittee. At the next trash monitoring work group, we will start talking about the formation of that group. If you want to stay informed, you can also go to the state email system. If you, there's the link, you can go to the general interest, click on that bar, and it'll just choose the trash monitoring work group. Finally, our next trash monitoring work group, where we're discussing the formation of this microplastics subcommittee, will probably happen around or on November 1st. I'm still waiting to hear back from 2 people on whether or not they are open on that day. And again, if you have any questions, please reach out to Scott or myself, and we will respond to you as quickly as we can. That's it for the webinar. If we want to thank all of our speakers. What a great group we had. And I will turn it over to Erick if you have any parting comments on where people can find the information.
Thank you so much for your participation. Thank you for all the wonderful information that was given. You guys are just fantastic. Thank you, Shelly. Thank you again for organizing this, and Scott for all your expertise in bringing this fantastic panel together. I look forward to seeing your subcommittee thrive.
Great. Thank you. Bye, everyone.
Thanks again, everyone.